Good morning, students. Today in the first class, we will begin with an introduction to recombinant DNA technology. If we look at the history of genetics, we have already studied about genetics in your class 11. If you remember the name of the father of genetics, what is the name of the father of genetics? Gregor Johann Mendel. He is the father of genetics because he laid the universal laws of heredity that you have all studied about, right? So genetics, the story of genetics actually begins with this person, Gregor Johann Mendel, who gave the laws of heredity to us. So this is what we call as the era, or you can simply say the period of classical genetics. Classical genetics, especially when his work was rediscovered in the year 1900, when his work was rediscovered, a rediscovery of Mendel's work. After this, many scientists about whom you have already studied, like Carl Correns, Bateson and Punnett, all these people did a lot of work in the field of genetics, and they were mostly trying to formulate more laws which would agree with the laws of heredity and which would improve these laws of heredity. So this period of genetics is called as classical genetics. Alright. So this period, it lasted for about 10 to 20 years, 1900 to 1910 or so. And after that came the we talk about the era of modern genetics. Again, you would have heard the name of this person, also called as the father of modern genetics. His name is, any guesses? He has worked with the organism Drosophila. Yes, you may be right. You might have guessed the name, T.H. Morgan. T.H. Morgan, who worked with the famous Cinderella of genetics, we call Drosophila the Cinderella of genetics. This person again did a lot of work, very important theories about which you have already studied, the theory of chromosomal inheritance. Then he has founded about linkage and crossing over and mutagens, and his student Stutterwand, he discovered that certain chemicals and radiations can be used to find out about can be used to create mutations and also to find out about the role of genes. The era of modern genetics goes from T.H. Morgan right up to Watson and Crick. What did Watson and Crick do? They found out the structure of double helix. Alright, the proving of DNA as the genetic material, and also the how the DNA is expressed, the genes in DNA is expressed. All these were studied during this period, 1910, 1920s, 30s, 40s, 50s, almost up to the end of 50s. We learned a lot of modern genetics wherein we understood the mechanisms of DNA expression, how DNA replicates, how DNA is able to make mRNA through the process of transcription, and protein through the process of translation. All this was done during the era of modern genetics. Okay. Now, at the end of 1950s and the beginning of 1960s, this is what scientists thought, that probably there is nothing more to study in biology. This is the end of genetics. This is the end of biology. We have understood everything that happens in biology. So this is it. There is nothing more to do with biology, or not much that can be done in biology, because we have understood all the molecular processes that are occurring inside the cell. Now, so the period of 1960s especially, it was basically it was a silent period, not much happening in the field of biology. But come 1970s, there were three important discoveries that took place. Three important discoveries took place in the 1970s. Now, what are these discoveries? Now, scientists were trying to find out, they understood that the material of our heredity, that is the hereditary material, is DNA. DNA consists of 4 nucleotides. That's it. So whatever is our gene is just a sequence of 4 nucleotides. So can we put a molecule of DNA, any DNA, into any cell? Is it possible? Any DNA into any cell? And can that DNA express inside that cell? So what were they trying to do? Take any DNA. If it goes into any cell, can it express inside that cell? Now, if you remember 1928, you've studied about this person called Griffith, who did an experiment on transformation. So what was this transformation? He used the causative organism, the organism that causes pneumonia, and he was trying to do something wherein he found out that by the process of transformation, the R strain can get converted into S strain. This is what he found out. And later on, it was proved that DNA from the the DNA from the S strain moves into the R strain, converting it into the S strain, the virulent S strain. This is the experiment that we have already studied in your 11th standard. Alright, so this experiment, the transformation experiment by Griffith, is what the scientists were trying to do over here. So is this only true with the DNA of S strain entering the R strain and converting it into S strain, or any DNA into any cell and then can it express in that cell? I hope you have understood this. Okay, so the scientists were not very successful in trying to make any DNA enter into any cell, and they were not able to express it. The problem they were mainly coming across, the problem with this was, number one, the DNA has to enter the cell. Enter the cell. So as you all know, the cell membrane is selectively permeable. It will not allow DNA to enter. Most of the DNA is discarded by the cell. So DNA cannot enter the cell. The second problem is, let us imagine the DNA somehow entered the cell, but the DNA was not replicating. So what will happen if we have a cell here? I am taking a bacterial cell, a small bacterial cell. Somehow this DNA that we want to enter the cell, it has entered into the cell. Alright, but when it is inside this cell, but when this cell is dividing, what happens? Only one of the daughter cell gets it. One of the daughter cell gets it, the other cell does not get it. Now see this, these two cells are dividing. So again one daughter cell gets, the other one doesn't get. This one obviously doesn't have that foreign DNA. Okay, so now you imagine like this, the cells go on dividing like this. Finally, if you have, let's say, 1,000,000 cells, you will have only one cell with the foreign DNA and all the other cells will not have the foreign DNA. So what has happened? Will you be able to find that one cell among these million cells? Obviously you will not be able to find it. So this is what was happening. The problem was that DNA was not replicating. Alright, so the scientists once again put a question. This is the foreign DNA. Foreign DNA. The cell also has its chromosomal DNA. This is also the chromosomal DNA. Both of them are made up of nucleotides. The 4 nucleotides, GATC. This is able to divide. Both the daughter cells get the chromosomal DNA. Alright, but why this DNA is not dividing? Why the foreign DNA is not dividing? So this was the question. Why the foreign DNA is not dividing? So once again they tried to make out the process of DNA replication. They tried to understand the process of DNA replication, and then they understood that for a DNA to replicate, if a DNA molecule has to undergo the process of replication, you have already studied this process of replication. I hope you have once again revised the process of replication. The process of replication begins at a particular point, if you remember, which is called as origin of replication. It's called as origin of replication, or simply it's called as ORI. What is this ORI? ORI is origin of replication. This is the point at which the DNA replication starts. So DNA replication starts at the origin of replication. If a DNA molecule has an ORI, let us imagine there is an ORI here. If the ORI is present, it will replicate inside the cell in which it is put into. If there is no ORI, it does not replicate. So this is what they understood. Okay, so if this ORI is there, DNA is able to replicate. No ORI, no replication. So now again the question came, so if I take any DNA molecule, let's say this is a DNA molecule which is of my interest. I'll draw a double-stranded DNA molecule. Supposing it has an ORI, and I put it into any cell. Will it replicate? Yes, it will replicate. So this is what they understood. So they started looking out for DNA molecules. Which are such DNA molecules with ORI? Which DNA has ORI? Which are the DNA molecules with ORI? They came across two candidates. One, plasmids, and two, bacteriophages. Bacteriophages, as you all know, are viruses that can infect bacterial cells, So how are they able to infect? We have already studied about this. So it has bacteriophage first attacks this bacterial cell. It inserts its DNA into the bacterial cell, degrades the bacterial DNA, and then it starts making copies of its own DNA. Alright, and it is able to replicate and so on. Alright, so if it is the meaning that it is able to replicate its DNA means that it has ORI. That is why this DNA is able to replicate inside the bacterial cell. You got it? So it means bacteriophages DNA already has an inbuilt ORI. You can think like that. Now coming to plasmids. What are plasmids? They are extra chromosomal DNA which bacteria have. Extra chromosomal, that means other than the chromosomal DNA. This is a chromosomal DNA. Other than this, there are certain small extra chromosomal DNA inside the bacterial cell. They are small compared to the chromosomal DNA. They are circular, double-stranded DNA, and they are capable of autonomous replication. Autonomous replication means they can replicate irrespective of whether the chromosomal DNA is replicating or not. This is the meaning of autonomous replication. So these two were suitable candidates, plasmids and bacteriophages, suitable candidates for DNA with ORI. ORI means origin of replication. So they thought, if we insert these two into the bacterial cell, they will be able to replicate inside the cell. Okay? So this is one of the things that scientists started understanding. So along with this, there were three important developments that took place in the 1970s. You can write this down in your copy also. Three important developments in the 1970s. The first one was scientists were able to do transformation of E. coli successfully. Transformation of E. coli. They learned how to do transformation of E. coli successfully. Transformation of E. coli means they learned or they discovered mechanisms by which foreign DNA can be inserted into E. coli cells successfully. So this mechanism they learned, and they were able to do this with a high rate of success. So this was one of the important developments that took place in the 1970s. Second important development that took place was they discovered how to cut and join. I'm sorry, there's some water there, so it's not writing. Cut and joining reactions of DNA. Cutting and joining reactions of DNA were discovered. And cutting and joining reactions of DNA means they found out mechanisms by which they can cut DNA wherever they want and they can join DNA wherever they want. And they also learned how to monitor the cutting and joining reactions. Cutting and joining reactions. They learned how to monitor the cutting and joining reactions. What is the meaning of monitoring? Monitoring means, see, if I take let's say I take this pencil and I cut this here. So I get two pieces of pencil. Will you be able to see it? Yes, you will be able to see it. But DNA, if I take DNA, which is just a macromolecule, and I say I've cut the DNA, I've joined the DNA, will you be able to see it? You will not be able to see it. So, just by telling you cannot believe it, you need proof in science. So, when we say monitoring the cutting and joining reactions, it means they found out mechanisms by which they could actually make out whether they have actually cut the DNA or they have actually joined the DNA and whether it has been successful or not. This is what they were able to do. So these three developments are very, very, very important for us biotechnologists, and for recombinant DNA technology. Transformation of E. coli, cutting and joining reactions of DNA, monitoring the cutting and joining reactions. Okay, so with those three developments, as I said, the era of recombinant DNA technology or biotechnology as we know it actually started. Okay, so let us first try to understand the meaning the steps in gene cloning. You've already written this in your copy. You can look into your copies as well. So these are the steps in gene cloning. Okay, so before we go into the steps, let us look into the definition. This is the definition of gene cloning or recombinant DNA technology: the process by which a DNA fragment is inserted or combined with another DNA molecule so as to create a new combination of DNA and insert it into a host cell in which does not already exist but in which it is capable of continuous propagation. Okay, so you don't have to worry about this big definition. Let us try to break it into pieces and understand. So it is a process. It is a process. So what are you going to do in this process? A DNA fragment is combined with another DNA molecule. So you're going to take one DNA molecule and you're going to combine with another DNA molecule. You're going to combine it together. And you're going to create a new recombinant DNA. It's called a recombinant DNA. Alright. So this is a new combination of DNA. Alright. And what are you going to do with this recombinant DNA? You are going to insert it into a host cell. So why is this called a host cell? Because now it is going to host this recombinant DNA. Or in short, we can call it rDNA. Insert it into a host cell. Alright, that is the next thing. And this is very important, in which it does not already exist. This recombinant DNA, you have created this. This is a new combination. It does not already exist in that host cell. Alright, so that's the important point. It does not already exist, but in which it is capable of continuous propagation. Propagation means replication. So when this cell divides, these daughter cells also will get the recombinant DNA. That is the meaning of gene cloning. I hope you have understood this. Now let us look into the steps. If you understood the definition, the steps are going to be very easy. Isolation of a DNA fragment having a gene of interest. So what will you do? The first step, you are